Hello, this is an introduction to taxonomic searches in OrthoMCL. I'm going to show some basics of the search method on OrthoMCL DB. But first, let me just explain what OrthoMCL is. OrthoMCL is a database of orthologous proteins. And by orthologs, I mean proteins that are the same gene in different species. They are similar in sequence and they have the same function. OrthoMCL uses sequence similarity in different species across all of UPathDB. So 650 eukaryotic pathogens, hosts, and vectors, plus an additional 149 taxonomically diverse core species to give our orthogroups breadth and also to enable searches that specifically leave out or include different groups of organisms. Recent gene duplications we call paralogs, and if they're quite recent and the sequences are similar and have similar function, they'll be included in the same orthogroup. If they're quite ancient, they may have diversified in function and in sequence and be split into different orthogroups. Due to sequence duplication, often there are many-to-many relationships between orthologs across different species. And so all of those orthologs might be contained in a single orthogroup, and it would be a many-to-many relationship, not a one-to-one relationship for the orthologs across the two different species. In OrthoMCL, we have some tools for data mining to find orthogroups, either with specific text terms, a PFAM ID, a certain number of sequences in the group, a certain number of organisms in the group. One of the most interesting search tools we have we call a phyletic pattern. It allows you to specify the taxonomic relationships of groups of organisms in a particular orthogroup. The phyletic search has two interfaces. The first one involves a mini taxonomic tree with all the organisms in OrthoMCL, and you have the ability to check on and off individual groups. You can open them up and it goes all the way down to the species level. And in some cases below the species level when we have more than one strain of a particular species. The simple kind of search we could do here is say, what orthogroups contain fungi, but no archaea and no bacteria. So we'll make the archaea and bacteria red, the fungi yellow, which means any species in fungi, the orthogroup must have at least one fungal species. How many orthogroups do we get from a search like that? 270,000. The result comes up really quick, but the table of the individual orthogroups takes a few seconds to load. There's a lot of information there, and it's very useful to browse the results. So we'll wait a moment for the result to come up. All right, so here's the 270 orthogroups. And if you scroll to the right, you see we have PFAM domains, EC numbers for enzymes, and this graphical representation of the different major groups and how many species are found in that orthogroup from that larger taxonomic group. So we look for fungi, but not archaea or bacteria. In some groups, there's a lot of metazoa, a lot of plants, etc. If we want to get more specific with our search, then we have to learn how to use the regular expression language, there's a tutorial here or a description of how the regular expression language works. There's a four-letter abbreviation for each taxa, which could be an individual species like Homo sapiens, or it could be for a large group like eukaryotes, an equal sign, and a number. The number would indicate the number of proteins. So we'd be looking for an ortho group that has at least 5 proteins that come from eukaryotes, or an ortho group that has 5 or more proteins from humans. You can also group these expressions with and or statements. So if we'd like to find an ortho group that is common in fungi, say that belongs to at least 10 fungal taxa, but not any archaea, not any bacteria, and perhaps not any um, yeah, well, actually, we'll just leave it like that and get answer. So these are common in fungi, but not in archaea or bacteria. Now we've lost a lot of the 270,000 orthogroups that we had before. Probably though some of those involved groups where maybe there was just one fungal uh, species, one fungal protein, perhaps even a singleton, a protein that was poorly annotated and doesn't show up in any other organism. But the interesting ones, the good ones that have lots of fungal organisms are here. So if we were really interested perhaps in finding a fungal gene, fungal protein that's not present in metazoa or not present in mammals, we could extend that regular expression and add more criteria. Another way to do this is to use a search strategy. So uh, this search right now is already in our search strategy system, and we have the ability to add a step. So in this step, we could add, say, a text term, and we could look for synthetase. We also like to add an asterisk to the beginning and end of our search term to get all the compound words. So it might be synthetase-like or alanine synthetase or something like that. And so if we add that on top of our search strategy, then we are combining a taxonomic search with a functional search. And that brings us down to 152 orthogroups that contain some sort of synthetase mentioned in any protein in the group and fits our taxonomic criteria of being present in fungi. All right, so actually that's all I wanted to say. That ends this tutorial.